Welcome to the Deep Dive, the show where we distill complex topics into the most important nuggets of knowledge, just for you. Today, we're plunging into one of life's most fundamental mysteries. How does information actually flow through living systems at the most basic cellular level? We're talking about neurons, synapses, and the incredibly precise dance of signals that makes everything from thought to movement possible. To kick off our deep dive, let's start with a truly captivating and frankly kind of deadly example, the tropical cone snail, Conius geographus. Picture this. It's small, it's slow, but it's an absolute terror for fish in its habitat. It's a carnivore that hunts, kills, and devours its prey with, well, astonishing speed. Its secret, a hollow, harpoon-like tooth. It injects venom, paralyzing its prey almost instantaneously. This venom is so potent, just one injection has unfortunately killed unlucky scuba divers.
It's really something. And what's truly astonishing about this snail's weapon is that its venom isn't just, one type of poison. It's a complex cocktail of toxin molecules. Each one is specifically designed to disable neurons, the very nerve cells responsible for transferring information throughout the body. When an animal is struck, it loses all neuronal control over its movement and even its breathing. Escape, defense, survival, utterly impossible.
It's mind-boggling how such a small creature can exploit this fundamental biological process, neuronal communication, to such devastating effect. It really highlights the incredible power and the precision of these signals.
And that precision is precisely what we're here to unpack today. Our mission in this deep dive is to explore the specialized structure of neurons, how they use both electrical and chemical signals. We'll break down how these signals are generated, transmitted, and interpreted. We want to help you grasp these fundamental biological concepts without getting overwhelmed by jargon.
Think of it as a shortcut, maybe. A way to get well informed about the very essence of how living things process information, drawing directly from, the insights of modern biology. Now let's get to the core of how this complex system is actually built. Imagine a neuron with me. At its heart is the cell body. That's the metabolic engine, right? Housing the nucleus and all the essential machinery.
Exactly. And from this cell body, you'll see these branched extensions. They look a bit like antennae, maybe. They're called dendrites. These are specialized to receive signals from other neurons. Then there's the long, slender axon. This acts like a transmission cable, sending signals away from the cell body towards other cells. And to give you a sense of scale, some axons, like those stretching from a giraffe's spinal cord all the way down to his foot muscles, can be over a meter long.
A meter. Wow, that truly puts the scale of these neural pathways into perspective. So how does a signal even maintain its integrity over such a huge distance?
It's incredible, isn't it? The signals that travel down the axon are typically generated right at its base, this cone-shaped area called the axon hillock. Then at the far end, the axon branches out, and each branch forms a specialized junction. We call that a synapse. The very tip of that branch is the synaptic terminal. Now, at most synapses, information gets passed from the transmitting neuron, that's the presynaptic cell, to the receiving cell, the postsynaptic cell, using tiny chemical messengers. We call these neurotransmitters.
Okay, so we've got these intricate parts. Dendrites, like antenna for receiving, a central engine in the cell body, and this long transmission cable, the axon. How does this all fit together to actually process information? It sounds like a beautifully complex machine.
It is, yeah. And it doesn't work alone. It relies on vital support staff. Glial cells, or just glia. The name comes from the Greek word for glue, because they literally help hold things together. And these remarkable cells actually outnumber neurons significantly, maybe 10 to 50 times more in the mammalian brain. Glia, nourish neurons, they insulate their axons for faster signaling. They regulate the fluid around them. Yeah. Sometimes they even get involved in information transfer themselves.
Okay, let's go back to our cone snail for a second. It's surveying its environment, looking for prey. How does its nervous system actually work to find and attack that fish? Are the basic steps the same as in, say, us?
They really are. Its nervous system, just like ours, processes information in three main stages. First, you've got sensory input. That's where sensory neurons, like the ones in the snail's siphon, detect external stimuli, maybe the scent of a fish. Second, there's integration. Here, interneurons, which form local circuits in the snail's brain or ganglia, they process and interpret that sensory input. They decide, okay, is a fish present? Where is it? And finally, there's motor output. This is where motor neurons transmit signals to muscle cells, like the ones that release the snail's harpoon, or maybe to glands, causing a specific response. And this whole process is organized into two main systems. The neurons doing the integration, they're often bundled into the central nervous system, the CNS, think brain and ganglia. The neurons that carry information into and out of the CNS make up the peripheral nervous system, or PNS. And when these PNS neurons are bundled together, we just call them nerves. It's a really neat division of labor.
Right, so it's clear neurons don't operate in isolation. They form these complex circuits, constantly passing information around. And this brings us to the ions, right? That crucial electrical part. How does a neuron maintain an electrical charge? It's not like they have a tiny battery stuck on them.
No, not exactly. But they function a bit like one, thanks to something called membrane potential. This is basically the voltage difference across the plasma membrane. The inside of the cell is negatively charged compared to the outside. Think of it as a form of potential energy, just sitting there, ready to be used. For a neuron that's not actively sending a signal, we call that a resting neuron. This resting potential is usually somewhere between -60 and -80 millivolts. Tiny, but crucial. And the key players in setting this up are ions, specifically potassium ions, K+, and sodium ions, Na+. Inside the cell, there's way more potassium. Outside the cell, way more sodium. These concentration gradients are absolutely vital, and they're actively maintained by a protein called the sodium-potassium pump. This pump is constantly working, using energy from ATP to shuttle 3 sodium ions out for every 2 potassium ions it brings in. It keeps those differences in place.
Okay, so the pump is always working, keeping those gradients, but you said it pumps positive ions both ways. Why is the inside so negative at rest? Where does that strong negative charge come from?
Oh, that's a great question. The pump sets the stage, but the resting potential itself is mainly due to specialized protein channels called ion channels. Think of them like selective gates in the cell membrane. In a resting neuron, many potassium leak channels are just always open. This allows K+ to continuously flow out of the cell following its concentration gradient going from high concentration inside to low outside. But crucially, very few sodium channels are open at rest. So you have this constant outward flow of positive potassium ions, while other negatively charged molecules like proteins are trapped inside. That net outflow of positive charge is what makes the inside negative. Now, as positive K+ leaves, the inside gets more negative, right? This negative charge starts to pull some K+ back in electrically. Eventually, you reach a balance point for potassium, where the chemical push out equals the electrical pull in. This balance point is called the equilibrium potential. The neuron's actual resting potential is very close to potassium's equilibrium potential because the membrane is so much more permeable to K+ than to Na+ at rest.
This seems so fundamental. It's not just about nerve impulses, is it? This whole business of moving ions across membranes must be happening all over the place in biology.
Oh, absolutely. You're spot on. Ion movement across membranes is, well, it's a universal activity in living things. It drives an incredible range of processes. Think about this. The same basic electrochemical principles that let your brain think deep thoughts are also helping a plant breathe, essentially.
Yeah.
In plants, guard cells pump out hydrogen ions, H+. This creates a voltage that drives potassium ions, K+ in. Water follows the potassium, the cells swell, and the little pores on the leaf open up for gas exchange. It's fundamental.
Wow. A universal language of ions and voltages. Okay, so we've got this neuron sitting at rest, charged up like a tiny battery, ready to go. How does it actually send the signal? What flips the switch from resting potential to, well, action?
Right. That's where gated ion channels come into play. Unlike those passive leak channels, these are like little switches or doors that open or close in response to specific triggers. And they dramatically change the membrane's permeability to certain ions. A key type we need to understand are voltage-gated ion channels. As the name suggests, they open or close when the membrane voltage changes. Now, when these gated channels open, they can cause 2 main types of shifts. If, say, gated potassium channels open, more K+ rushes out. This makes the inside even more negative. We call this hyperpolarization. It makes it harder for the neuron to fire. But if gated sodium channels open, Na+ floods in. This makes the inside less negative, moving the voltage closer towards 0. This is called depolarization, and this is what can lead to a signal.
Okay, depolarization brings it closer. But are all depolarizations a full-blown signal, or is it more like a dimmer switch versus an on-off switch?
That's a good analogy initially. We have what are called graded potentials. These are local, temporary shifts. Their size or magnitude depends on how strong the initial stimulus was. But they're like ripples, they fade out over distance. So they're okay for short-range communication, but not for sending a signal down that long axon, like the one of the giraffe's leg. But if a depolarization is strong enough, if it reaches a critical level, then something truly spectacular happens, an action potential. This is a massive, brief, all-or-none electrical signal. All or none means it either happens fully or not at all. There's no halfway action potential. It's triggered when the depolarization hits a certain threshold, often around -55 millivolts in mammals. Once you hit that threshold, it sets off this incredible positive feedback loop. Voltage-gated sodium channels snap open, Na+ rushes in, causing more depolarization, which opens even more sodium channels. It's a cascade, an explosive event that rapidly flips the membrane potential. Whoa, okay, that sounds like a really fast, dramatic dance of ions. Can we walk through that?
Yeah.
Visualize the whole sequence? Absolutely. Let's picture those voltage-gated channels again. Stage 1, resting state. All the main voltage-gated sodium, Na+, and potassium, K+ channels are closed. The membrane is at its resting potential. Stage 2, depolarization. A stimulus arrives. Open some voltage-gated Na+ channels. Sodium starts to trickle in. If this trickle is enough to push the membrane potential to that threshold level. Stage 3, rising phase. Bang. Most of the voltage-gated Na+ channels rapidly open. Sodium ions flood into the cell. The inside zooms from negative to positive, maybe reaching +30 or +40 milliVs. Stage 4, falling phase. This happens almost immediately after. The voltage-gated Na+ channels have this little inactivation gate that slams shut, blocking further Na+ entry. At the same time, the slightly slower voltage-gated K+ channels open up. Potassium ions now rush out of the cell, bringing the potential rapidly back down towards negative. Stage 5, undershoot. The K+ channels are a bit slow to close. So much K+ leaves that the membrane briefly becomes even more negative than the resting potential, getting closer to potassium's equilibrium potential. Then finally, the K+ channels close, the Na+ channels reset, the inactivation gate opens, but the main gate stays closed, and the membrane returns to its resting state. The whole thing takes just a few milliseconds. And a really key part of this process is the refractory period. This is a very brief moment right after an action potential fires when the neuron cannot fire another one, mainly because those sodium channels are inactivated.
Ah, okay. And that's why the signal only travels in one direction down the axon, right? It can't double back on itself.
Precisely. The refractory period ensures the waves of depolarization moves forward, away from the cell body, towards the axon terminal. It's a brilliant piece of biological engineering for one-way traffic, and it also relates to how information is coded. A stronger stimulus, like a brighter light or a louder sound, doesn't create a bigger action potential. Remember, they're all or none. Instead, a stronger stimulus causes more frequent action potentials. The frequency encodes the intensity.
It's really mind-boggling to think that just a tiny flaw, a mutation in one of these little ion channel proteins, could cause something like muscle spasms or even epilepsy. It just highlights how incredibly fine-tuned this whole system is.
It really does. And sadly, yes, mutations in genes coding for ion channels can cause significant disorders. You mentioned myotonia, where mutations in muscle sodium channels cause stiffness and spasms. Or in the brain, mutations in sodium channels can lead to some forms of epilepsy, where neurons become hyperexcitable and fire in uncontrolled bursts, causing seizures. It really underscores their critical role.
Okay, so the signal is reliable, it goes one way, but sometimes our bodies need to react fast. Think about pulling your hand away from something hot. How has evolution optimized this system for sheer speed?
Great question. Evolution's tackled this in a couple of main ways. One simple strategy is just making the axon wider. Like a thicker pipe allowing more water flow, a wider axon offers less internal resistance to the flow of ions, so the action potential propagates faster. We see this in invertebrates like squid. They have these famous giant axons, sometimes up to a millimeter thick. This allows for super fast escape reflexes, maybe reaching speeds of 30 meters per second.
Wow. But a millimeter thick, that takes up a lot of space, doesn't it?
It does. Which is why vertebrates, including us, evolved a much more space-efficient solution. The myelin sheath. This is essentially electrical insulation wrapped around the axon. It's formed by glial cells, Schwann cells in the peripheral nervous system, oligodendrocytes in the central nervous system. They wrap layers and layers of their fatty membrane around the axon, kind of like insulation on an electrical wire. But the myelin isn't continuous. There are these small gaps called the nodes of Ranvier, where the axon membrane is exposed, and that's where the voltage-gated sodium channels are concentrated. So instead of flowing smoothly down the axon, the action potential effectively jumps from one node to the next. This process is called saltatory conduction, from the Latin word saltare, meaning to leap.
Leaping signals, that sounds much faster.
It is much, much faster in conduction in an unmyelinated axon of the same diameter. And it's incredibly space efficient. You could fit maybe 2,000 myelinated axons in the space one of those squid giant axons takes up. It's a key vertebrate innovation.
Okay, so the action potential has zipped down the axon, maybe leaping from node to node at incredible speed. It reaches the end, the synaptic terminal. Now what? How does that electrical signal get passed on to the next cell? It can't just loop across empty space, surely.
You're absolutely right. It generally can't just jump the gap. This critical transfer point is the synapse. And the most common type, the one that allows for really complex information processing, is the chemical synapse. Okay, let's visualize this again. It's like a tiny, incredibly fast chemical conversation. First, the presynaptic neuron, the sender, makes these chemical messengers, the neurotransmitters, and packages them into little bubbles called synaptic vesicles. When the action potential arrives at the axon terminal, it causes a depolarization there. This change in voltage opens up special voltage-gated calcium, Ca2+, channels. Calcium ions, which are much more concentrated outside the cell, rush into the terminal. This influx of calcium is the key trigger. It causes the synaptic vesicles loaded with neurotransmitter to fuse with the presynaptic membrane, and poof, the neurotransmitters are released into the extremely narrow gap between the neurons and the synaptic cleft.
How narrow are we talking here? Really tiny.
Incredibly tiny, less than 50 nanometers wide. That's smaller than the wavelength of visible light. Diffusion across this gap happens almost instantaneously. Once across, the neurotransmitters bump into and bind to specific receptor proteins embedded in the membrane of the postsynaptic cell, the receiver. This binding drives a response in that receiving cell. And then, very quickly, the signal needs to be turned off. The neurotransmitters are cleared from the cleft. They might just diffuse away, or they might be actively pumped back into the presynaptic neuron or nearby glial cells, or they might be broken down by enzymes right there in the cleft.
That clearing process sounds crucial. What happens if it goes wrong? Are there real-world, maybe even frightening consequences?
Absolutely devastating ones, potentially. Think about nerve gases like sarin. Sarin works by inhibiting acetylcholinesterase, the enzyme that normally breaks down the neurotransmitter acetylcholine in the synaptic cleft. If acetylcholine isn't broken down, it keeps stimulating the postsynaptic cell, like muscle cells. This leads to continuous muscle contraction, paralysis, including the respiratory muscles, which is why it's lethal. It highlights how critical that rapid off switch is. But on a more positive note, the fact that chemical synapses can be modified, changing how much neurotransmitter is released, or how sensitive the postsynaptic cell is, that plasticity is thought to be the fundamental basis for things like learning and memory.
Fascinating. So it's mostly these chemical handshakes. Are there any direct electrical connections?
Yes, there are. Less common, but important in specific circuits are electrical synapses. Here, the membranes of the two neurons are basically connected by channels called gap junctions. Ions can flow directly from one cell to the next, meaning the electrical signal passes almost instantly. These are super fast and reliable, but they don't offer much opportunity for modification or complex processing. You find them in pathways where speed and synchronization are paramount, like in some escape reflexes or coordinating heart muscle contractions. Okay, back to the chemical synapse. When the neurotransmitter binds on the postsynaptic side, what exactly happens? You mentioned a response, right? Often, the receptor protein itself is an ion channel. We call these ligand-gated ion channels or ionotropic receptors. Ligand just means a molecule that binds to another, in this case, the neurotransmitter. When the neurotransmitter binds, the channel opens, ions flow through, and this causes a small temporary change in the postsynaptic membrane potential. We call this a postsynaptic potential, or PSP. Now, if the channel lets positive ions like sodium in, causing a depolarization that brings the membrane closer to the threshold for firing an action potential, we call it an excitatory postsynaptic potential, or EPSP. But if the channel lets potassium out, or negative ions like chloride in, making the inside more negative, hyperpolarization, and moving it further from threshold, we call it an inhibitory postsynaptic potential, or IPSP.
Okay, EPSPs are like a little go signal, and IPSPs are like a stop or slow down signal. But you said they're small and temporary, so a single EPSP usually isn't enough to make the postsynaptic neuron fire its own action potential, right? How does it decide? It must be doing some kind of calculation with all these incoming signals?
It absolutely is. Neurons can receive input from hundreds, even thousands of other neurons simultaneously, both excitatory and inhibitory. The neuron's decision center is typically the axon hillock, that spot where the axon joins the cell body. The axon hillock constantly integrates or sums up all the incoming EPSPs and IPSPs. This is called summation. There are two main types. Temporal summation is when you get multiple EPSPs from the same synapse firing in rapid succession. They arrive so close together that their effects add up, potentially reaching threshold. Think summing over time. Then there's spatial summation. This is when EPSPs arrive simultaneously from different synapses located at different spots on the neuron. Their effects also add up. Think summing over space. And of course, IPSPs are being summed too. They counteract the EPSPs. So it's this constant dynamic interplay. If the grand total of depolarization at the axon hillock at any given moment reaches threshold, then the postsynaptic neuron fires its own action potential down its axon. If not, it stays quiet. And it gets even more sophisticated.
Hmm.
Not all neurotransmitter receptors are direct ion channels. Some are linked to signal pathways inside the cell. These are called G protein-coupled receptors or metabotropic receptors. When a neurotransmitter binds here, it activates a sequence of events involving other molecules, like G proteins and second messengers. These pathways can then indirectly open or close ion channels or even change gene expression. These effects are generally slower to start than the direct ligand-gated channels, but they tend to last longer and can have broader, more amplified effects within the cell.
Wow, it's layers upon layers of complexity. And you mentioned there are lots of different neurotransmitters doing these jobs. Can we touch on a few key ones just to get a feel for the variety?
Sure. There are maybe over 100 known or suspected neurotransmitters grouped into different classes. A really major one is acetylcholine. It's vital at the junction between motor neurons and skeletal muscles, causing them to contract. But it also plays key roles in memory and learning within the brain. Interestingly, acetylcholine can be excitatory at one synapse, like muscle, and inhibitory at another, like in the heart, where it slows the heart rate. Its effects are terminated by an enzyme called acetylcholinesterase, the one targeted by sarin gas.
And drugs like nicotine affect it too, right?
Exactly. Nicotine mimics acetylcholine at certain receptors. And Botox, or botulinum toxin, actually prevents acetylcholine release, causing paralysis, which is why it smooths wrinkles, but is also highly toxic. Then you have simple amino acids acting as neurotransmitters. Glutamate is the workhorse. It's the most common excitatory neurotransmitter in the central nervous system, crucial for learning and memory. On the flip side, GABA, gamma-aminobutyric acid, and glycine are the major inhibitory neurotransmitters in the CNS. Many anti-anxiety drugs like Valium diazepam work by enhancing the effects of GABA, basically calming down neural activity. We also have biogenic amines derived from amino acids. This group includes things like norepinephrine, which is generally excitatory, and importantly, dopamine and serotonin. These are heavily involved in regulating mood, sleep, attention, and learning. Imbalances are linked to major conditions. Parkinson's disease involves the death of dopamine-producing neurons, while many antidepressants, like Prozac, work by boosting serotonin levels. There are also neuropeptides, which are short chains of amino acids. Examples include substance P, involved in transmitting pain signals, and endorphins, our body's natural painkillers. Opiate drugs like morphine mimic endorphins, and believe it or not, even some gases act as neurotransmitters. Nitric oxide, NO, is a fascinating one. It's not stored, it's made on demand and just diffuses to nearby cells. It's involved in things like penile erection. Viagra works by prolonging the effects of NO. Even carbon monoxide, CO, can act as a signal in the body.
That's an incredible diversity of chemical messengers, each with specific roles and receptors. Okay, so we've journeyed pretty deep into neuronal communication today, from that deadly cone snail all the way to the molecular dance at the synapse. Let's try and boil down the essential takeaways.
Sounds good. So at its core, we saw that neurons have these specialized structures. Dendrites receive signals, the axon transmits them, often over long distances, and synapses pass the signal on, usually chemically. We learned that the resting potential, that state of readiness, is actively maintained by the sodium-potassium pump and the selective permeability of the membrane, especially to potassium leak channels, creating an electrochemical gradient. Then action potentials are the all or none electrical signals that travel down the axon. They're triggered when the membrane depolarizes to threshold, involving that rapid opening and closing of voltage-gated sodium and potassium channels. Speed is boosted by wider axons, or more efficiently in vertebrates, by myelin and saltatory conduction. At the synapse, typically chemical, an arriving action potential triggers calcium influx, leading to neurotransmitter release. These messengers cross the synaptic cleft, bind to receptors, and cause either excitatory, EPSP, or inhibitory, IPSP, potentials in the postsynaptic cell. And finally, the postsynaptic neuron integrates all these incoming signals through summation at the axon hillock. If the net depolarization reaches threshold, it fires its own action potential. The wide variety of neurotransmitters and receptors allows for incredibly complex and nuanced communication.
It really is stunning. As we wrap up, just think about this for a moment. How incredible is it that everything we experience, our thoughts, feelings, memories, movements, it's all fundamentally built upon these incredibly rapid, precise, yet fundamentally simple dances of ions and molecules across tiny cell membranes. It's just a staggering testament to evolution's ingenuity that this intricate biological machinery operates pretty much seamlessly within us every second of our lives. What new possibilities might really understanding these fundamental lines of communication unlock for our future, treating neurological diseases, maybe even enhancing cognition?
That's a fascinating thought. It raises another question. If these basic electrical and chemical signaling mechanisms are so remarkably similar across such diverse organisms, from a simple snail to us, what does that tell us about the universal principles of how life processes information? Could understanding these biological circuits even give us deeper insights into computation itself, perhaps even artificial intelligence? Lots to think about.
Definitely food for thought. We hope this deep dive has given you a clearer, more tangible picture of how your own brain, and indeed all nervous systems, actually communicate. Thank you so much for joining us on this exploration.